Hello dear students. So in this video we are going to learn the part second of section 7 ICMR guidelines. Section 7 is the clinical trials of drugs and other interventions. So in the previous video what we have learned, we learned about first that's the guidelines general guidelines general guidelines for carrying out the clinical trials. Then second we learned about uh, the clinical trials for specific interventions. The first intervention was that's clinical trials for drugs and vaccine. Drugs and vaccine. Under this topic we learned uh, about the phases of phase of clinical trials, then the ethical consideration for different phases of clinical trials. Now in this video we will learn about the part 3 of this section 7, that is bioavailability and bioequivalence. Bioavailability and bioequivalence. Bioavailability and bioequivalence. This I have completed. I will mark it as. Then this we have also completed. So bioavailability and bioequivalence we will learn in this video. So first, ICMR, that's 2017 guideline module. They have given the definition for bioavailability. What is bioavailability? And they have also defined what is bioequivalence. Bioequivalence. Regarding bioavailability, they have mentioned it that bioavailability is a measurement of the proportion of the total administered dose. See, this is strict as per these guidelines. Whatever they have written in this module, I will be strictly delivering the lecture strictly with reference to this module. Whatever they have written, the definition, whatever they have written, here will be no add-on from my side. Okay, so here they have written bioavailability as a measurement of proportion of total administered dose of a therapeutically active drug, of a therapeutically active drug that reaches the systemic circulation, and therefore is available at the site of action, at the site of action. So this is the definition they have provided. So what's the definition here? So this is the total amount, or we can say the measurement of the proportion of total administered dose of therapeutically active drug that reaches the systemic circulation and is available at the site of action. Then bioequivalence, what they have defined is bioequivalence. So bioequivalence, they have defined it as, it's a term used in pharmacokinetics. It's a term used in PK, pharmacokinetics, when there are two or more medical products. When there are two or more medicinal products, these medicinal products, it can be either the proprietary preparations of a drug, containing the same active ingredient, containing the same active ingredient, containing the same active, they have mentioned it not here ingredient, they have mentioned it substance, active substance, that need to be compared for in vivo for biological equivalence, that need to be compared, that need to be compared in vivo biological equivalence. So this is the definition they have given, and these comparative studies are used to assess if the new version like generic produces the same concentration in the systemic circulation when given to human participants. If the two products are said to be equivalent, it means they will be expected to be for all intended and purposes the same. So just simple, if you don't want to remember the exact things what they have mentioned in this guidelines, like the definition all and this, what we have to follow strictly is the guideline. What when we carry out the clinical trials or when we carry out the clinical research, what they have mentioned about the guidelines, that guidelines we have to strictly follow. But definition, it's, we know that bioavailability is like if we give it a simple definition for bioavailability, it's a rate and extent of, it's a rate and extent of a drug that reaches to a systemic circulation. Simple definition, that reaches systemic circulation means rate is, it defines the time, okay, rate, and extent is amount, how much. For example, if we have taken the 100 mg of tablet and from 100 mg 80 mg of drug is in the systemic circulation. So what is the extent? Extent is 80 mg, and rate is, for example, it takes like 30 minutes. So what's the bioavailability? That's 80 mg. 80 mg. So this is a simple, it's a rate and x. If we give the simple definition, it's the rate and extent of drug that reaches the systemic circulation. And bioequivalence, in the simple terminology, we can say bioequivalence is, we can say, different pharmaceutical products which contain same API, active pharmaceutical ingredient. So these are the different pharmaceutical products which contain the same API, but may differ in like, they may differ in the dosage, like dosage form, like for example tablet, capsule, okay. And if we see, they can be, see, these bioequivalents can be of different types, like for pharmaceutical equivalent, where everything is same, like preparation is the same, active pharmaceutical ingredient is same, and excipients are the same, but the different is like different brands, okay. Then we have chemical equivalent. So these, we know all about these things, we don't have to discuss on this. We will follow what the guidelines here is for bioavailability and bioequivalence studies. So these are bioavailability and bioequivalence studies. They are actually used for to get the access, like for new formulations like generic formulations. When we compare the bioavailability of different formulations, and then we compare it with also generic drugs. For example, we have, this is any drug manufactured by any company. DX is any drug. Let's suppose DX is any drug manufactured by any company like Dr. Reddy, and TX is also the same. TX is also the medicinal product containing the same active drug as DX. Then, because these drugs, these are the brand drugs, that's the cost and it will be, usually costly. These drugs are expensive. And when we compare these two drugs, if the bioavailability of the both drugs are approximate same, then we need to compare also with the generic drug. And the bioavailability of generic drug is computed with these formulations, and the bioavailability of these formulations, like brand formulations, it should be almost or approximately same as the generic drug. Then this generic drug is, we can say, this is approved, and this can also get marketed. Now the important point is regarding the ethical issues, ethical considerations for bioavailability and bioequivalence studies. So ethical considerations for bioavailability and bioequivalence studies. So first, they have mentioned that all bioavailability and bioequivalence studies, they should be scientifically sound. They should be scientifically sound. They should be scientifically sound and ethical. They should be scientifically sound and ethical. And the ethical considerations are same. The ethical considerations here are same as discussed in phase first. And this ethical conduct of bioavailability, bioequivalence studies requires evaluation of the second point they have given, that the study should be assessed. The study should be carried out on the basis of risk-benefit assessment. Risk-benefit assessment. When the benefit overweights the risk, then the study should be carried. Then the third point they are mentioning is that and bioavailability and bioequivalence studies, it is carried out in healthy individuals, healthy volunteer individuals. This is actually a point to be noted here, that someone may ask you, like your interviewer and other different Viva team, they may ask you like bioavailability and bioequivalence study is carried out in which individuals? It's carried out in the patients, it's carried out in the healthy volunteers. This can be an important question. So the answer is that's healthy volunteers. And the point here is, and here in bioavailability and bioequivalence studies, there is actually no direct benefit to the participants, like therapeutic benefit. There is no therapeutic benefit to the participants. So there is no therapeutic benefit to participants. But the risk is here, like the participants may get some kind of adverse drug reactions. So the researcher, they should therefore put all the they should make a protocol for carrying out the all safeguards, to protect participants, And also the fourth guideline is that ethical committee must carefully review the recruitment method, payment for the participants, and consent procedure. And then also the last thing they have mentioned is that the amount of blood drawn from the bioavailability samples, or we can say sample or blood, the sample or blood which are drawn for bioavailability and bioequivalence studies should be within the physiological limit. They should be within the physiological. They should be within the physiological limit. Physiological limit means that unnecessary sampling is not here permitted. It's not ethically good. Okay. So this is all about the bioethical considerations for bioavailability and bioequivalence studies. First point is that you know what's bioavailability. Bioavailability is the rate and extent of drug rate and extent of a drug that reaches to the systemic circulation. This is bioavailability, and bioequivalence is a comparative study. So here bioequivalence, it is a comparative study which compares the bioavailability of different formulations containing the same active ingredient, or we can say containing the same pharmaceutical active ingredient. So the bioequivalence is the comparative study, and by studying this bioavailability and bioequivalence, we can get also the access, or we can make this is actually the surrogate for the generic product, for the development of generic formulations. And so the generic drug, it should have approximately same bioavailability as compared to that standard drug. That's the branded drug. Okay. So we know what's bioavailability. We know the bioequivalence definition. First, they have given the definition. Then they have given that's the ethical considerations, how we can carry out this bioavailability and bioequivalence studies. So they have mentioned that all bioavailability and bioequivalence studies, they should be scientifically sound and should be carried out with due ethical considerations as described in the phase 1. So whatever the ethical considerations they have given in the phase first study of the drug, that's phase first of clinical trials, same considerations we have to follow while carrying out the bioavailability and bioequivalence study. And they have mentioned that the study should be evaluated and should be carried out after looking the risk benefit assessment. If benefit overweights the risk, then we can carry out the study. And they have also mentioned that this is very important point, is that bioavailability and bioequivalence studies is carried out in the healthy voluntary individuals. And as in case of bioavailability and bioequivalence study, here is actually we are not our intention is not the therapeutic benefit. Our intervention is actually that's the bioavailability. So here we don't have any therapeutic that's the intention, but the risk the participants can get is that's ADR from the drug. So we have to take the safeguard to protect the participants. And they have also mentioned that is the ethical committee, they must carefully review the recruitment of the participants and the compensation for the participants, like the payment for the participants and the consent procedure. And they have also mentioned that the sample or blood which are done for bioavailability and bioequivalence, they should be within the physiological limit. They should not be like unnecessary samples should be not done, and which is actually non-ethical. So these are the ethical considerations they have given the ICMR 2017 guideline they have mentioned. So you can take the screenshot, then we will discuss the next thing. Then the fourth here is that is ethical implication of the study design. That's the fourth subsection, you can tell, fourth subsection under the section 7. Clinical trials of drugs and other interventions, they have given in the different parts, you can say. First the part is, as I'm again and again repeating, because so that chronological, you can remember it chronologically. So first is general guidelines, then the where they have mentioned the definition of clinical trials and guidelines for carrying out the clinical trials. The second topic, that's the clinical trials for drug and vaccine development. Then the third that we discussed today for a while, that's the bioavailability and bioequivalence. And the fourth, they have given there's the ethical implication of the study design. Ethical implication of a study design. You know, the clinical trials is having a wide range of methodological approach. So the ethical committee need to look the details of that's the ethical concern which are involved in the methodology, and they should look thoroughly the ethical concerns which are involved in the methodology, and should also discuss and should be transparent to discuss it with the researchers or the ethical concerns. Then you know that review they can tell they can give the expedited review, they can give the full review, that we will learn So first is that the clinical trial, you know that it's having a wide methodological approach, different methodological approaches. So the ethical committee, they needs to look at the different aspects of clinical concerns which are involved in the methodological methodology, different methodology. And says if there is serious adverse event, that serious adverse event which occur in the blinded study, then and this blinded if this serious adverse event occurs in blinding study, and which is then and it's important, it's very important in the interest of managing this and it is very important in the interest of managing this serious adverse event, unblinding mechanism should be available to research. And placebo, they have also given that a placebo may be used as a comparative under the consideration given in the box. If the researcher is using a placebo, that's the dummy medication, and then they should use under the following conditions. The placebo can be used in the clinical trial under these conditions. The conditions they have mentioned, that is whenever there is no established therapy is available, then we can use the placebo. And if withholding established therapy, if we withhold if there is established therapy and we withhold this established therapy, because this established therapy withholding established therapy does not pose any kind of serious harm or any harm to the participant, and if the disease is self-limited, and if there is established therapy but this established therapy does not yield any scientific reliable results, and placebo used does not add any harm. And they have given that a placebo may be used for a scientific reason. If we use a placebo, placebo is needed to be used for a scientific reason, then there should be some precautions which should be carried out during the clinical trial. Firstly, the protocol. The protocol should mention it. The protocol must add the safeguards for the participants, and there should be use of add-on trial design. That's add-on trial design we should use. Add-on trial design where the IP, that's the investigational product or placebo, are added to the standard care. Okay, so add-on trial design means where the IP, that is investigational product or a placebo, they can be added to the standard care. Expose placebo to few patients, 2:1 randomization. This is called unbalanced randomization. So this guideline, it means that we can give placebo to lesser number of patients and the IP to, that's so IP investigation product can be given to the patient to more number of patients, or we can say, yeah, and this placebo can be given, for example, 2:1 randomization, where the IP, where the two patients receive that's the IP investigation product, and one will receive, that is a placebo. So expose placebo to few patients and investigational products too. Then it means that we can use this investigational product we can give it higher in participants as compared to placebo. That's the 2:1 randomization. This is called unbalanced randomization. And we can also use the active comparator as additional. For example, any other effective therapy is there, then we can use 2:2:1. 2 is IP, two participants are receiving IP, 2 are receiving active comparator, and 1 is receiving placebo. And we have to ensure the translation to standard care, and after the post trial we have to give the standard care. And see, blinding, blinding means, blinding, it is of three types. One is single blinding, double blinding, and triple blinding, I think. 1 minute. Yeah, triple blinding. So open, and here I will write it. First one is open label. Open label is when there is no blinding. Then single blinding, double, and triple blinding. Open label means there is no blinding. The researcher, the patient, they knows that what the product or what the investigation product they are taking. And single blinding means when the participants or patient do not know what kind of, whether they are taking the IP, that's investigative product, or placebo. Double blinding means both the patient and the researcher don't know. Triple blind means researcher, patient, and analyst don't know. They all don't know which product the patient is getting. Either it's placebo, or it is the IP. So these are the types of blinding. Okay. So this is the ethical implication of study design. So the ethical implication of study design, the ICMR is here giving about this. Here it's the ICMR is mentioning about the methodological approaches for in study design, and in what conditions the placebo can be used, and placebo can be used as a comparator, and what are the precautions when the placebo is used, what are the precautions we have to take. So in this section, in this subsection, the ICMR is focusing on it. That's the when the placebo can be used as a comparator, and what are the precautions. That's all. So you can take a screenshot of this, and we'll discuss the next topic. Then the next topic here the ICMR have discussed is that's multi-centric trials, 7.5. So multi-centric trials, the multi-centric trials are carried out with the primary aim, that's the sound basis of the subsequent generalization of its results. And the ethics committees of all sites, they should follow the guidelines, and the trial should be carried out in accordance with these guidelines. And the ethical review procedure, that's the ethical review procedure for the common review, is given in the section 4.10. So these are the things which ICMR mentioned here. And then the next they have discussed that the phytopharmaceutical drug. The DNC drug and cosmetic rules 8th amendment of 2015 have defined it. That's a phytopharmaceutical is a purified and standardized fraction. Purified and standardized fraction which defined minimum 4 bioactive or minimum 4 bioactive or phytochemicals qualitatively and quantitatively assessed of an extract of medicinal plant, which can be used for external or internal, which can be used for internal or external use of human being or animal for diagnosis, treatment, mitigation or prevention of any disease or disorder, but does not include administration by parental route. This is important. Does not include parental administration. Okay. And all the details described in 7.2 here, this clinical trial for drug and vaccine, they also apply for this section, also subsection. So you can take a screenshot, then we will discuss the next topic. Next, they have discussed about the device trials. So here, device trial, that's they have defined a medical device. It's defined as a medical tool which does not achieve its primary action by pharmacological means or by any means like metabolic actions. And this medical device, it can be apparatus, implant, used for detection, diagnosis, prevention, treatment, sporting, and consumption. The ethical consideration is that the clinical trial for medical device, it should be carried out as per the Indian Good Clinical Practice Guidelines, and safety data of medical devices should be obtained in animals also before initiating the clinical trials in the human. And procedure to introduce medical devices into the humans and its safety, it should be also evaluated. If the participants wants to withdraw, though the medical device is internal, the medical device, for example, if the medical device is inserted internally and the participant wants to withdraw, the withdrawal should be accepted with respect to post-trial obligation should be emphasized with sponsor, and there should be follow-up for any that's the long-term adverse effects. They have also mentioned it like they have also mentioned here They have also mentioned that the device could be internally or externally for diagnosis, treatment, mitigation, or prevention of a disease or disorder, and depending upon the risk involved in devices. So depending upon depending upon the risk of devices, medical devices, they have classified it into they have classified it the first class is A, then second is B, C, and D. Though they have classified it like class A, class B, class C, class D. Class B is, this is based on the risk. Here if you see the risk for class A medical devices, the risk is low. For class B, risk is low to moderate. For C is moderate to high, and for D is, this is high. And if you look the examples here, class A, like class A medical devices like what? Thermometer, bandages, tongue depressors. This is the class A. Then the class B is like hypodermic needles. See the example, hypodermic needles or suction pumps, or we can say suction equipment. Class D is lung ventilator, and class D is, that is the heart valve. So they have classified these medical devices. And they have also written that devices used for in vitro diagnosis. For example, if the device is used for in vitro, they can be like in vivo diagnosis. Devices can be used for in vivo and also in vitro. These are like in vivo means with when we use it along with the human, either it's internal or it's external. In vitro, this can be in vivo. Okay, in vitro means when it is like it's not in contact with the human participants. That can be that or the in vitro. Okay, so they have mentioned that device used for in vitro diagnosis could be reagent, calibrator, control material, kit, instrument, apparatus, equipment, system, or specimen receptacle, whether used alone or in combination with any other such devices. This is intended by its manufacturer to be used in vivo for examination. Okay, so they have that simple. The thing they are trying to understand is that the devices, it can be two types. Medical device, it can be two types. First one, medical devices, it can be used along with the human participants. It can be used internally or externally for the diagnosis, mitigation, treatment, prevention, supporting conception, whatever it is. Okay, and then second is in vitro diagnosis. Like we can take the blood samples. It can be blood samples for the diagnosis, like kits. Okay, and reagent. And they have also mentioned that then. So this is the in vitro and in vitro diagnosis, and they have also defined that the medical devices, it can be notified, and non-notified. So notified are in vitro diagnostics. In vitro diagnostics for testing HIV, HCV, and blood grouping. And non-notified are those for testing malaria, TB, dengue, chikungunya, typhoid, syphilis, etc. Okay, so this is all about they have mentioned about the medical devices. Next, they have discussed about biologicals and biosimilars. Like here they have defined that biologicals or biopharmaceutical drugs, they can be composed of sugars, proteins, enzymes, nucleic acid, living cell or tissues, and produced with or without organic technology. The ethical considerations here they have discussed is that all aspects described in section 7.1 are also applicable here, and special care taken to review all the data generated, and the study should be carried out on the basis of risk benefit assessment. And if the clinical trial study the clinical trial study for biologicals and biosimilars are carried out, then the preclinical and bioassay data should be obtained before carrying out these clinical trials. So this is all about. Then we will see the next. Next, they have discussed about the clinical trial with stem cell. Clinical trial with stem cell. First, they have discussed that they have written that the clinical trial on stem cell, they are categorized into 3 classes based on the source and degree of risk to human participants. First one is permissible, like adult and cord blood. Then second is restricted, example is embryonic, and third is prohibited, that's reproductive cloning. In India, only this permissible and restricted clinical trials are carried with respect to associated with stem cell research. And to address issues associated with stem cell research, ICMR and DBT have published guidelines for stem cell research and therapy in 2007, 2013, 2017. And accept hemopoietic stem cell transplant. Accept hemopoietic stem cell transplant for hematological disorders. Any other use or categorized as a research and must be conducted as a clinical trial. Then these clinical trials should be approved by ethical committee and National Apex Center for Stem Cell Research and Therapy. And these clinical trials should be carried out in GCP, GLP, and GMP guidelines as per these guidelines. And all clinical trials must be approved by ICMR and National Apex Committee Stem Cell Research Transplant, and should be also clinical trials should be also approved by should be also approved by Clinical Trial Registry of India. It should be also. Okay. The next we will discuss the next topic. Next is they have discussed about surgical interventions. And about surgical interventions, the ICMR is the ICMR 2017 guidelines. They are emphasizing that surgical interventions that are being studied systematically must be considered as a research, and all general principles described in these guidelines. Like so, the ICMR is telling that any surgical intervention that are being studied systematically, which is not established, any surgical intervention that is not established but is being studied, they must be considered as a research and should be carried out and should be followed all the principles described in the guidelines. So the first guideline they have mentioned that the protocol where an established surgical intervention is to be studied, the researcher must provide a reference for the procedure and describe the most likely complications in the protocol for the ethics committee to review and perform risk-benefit assessment. The frequency of each complication should be also mentioned. And then next to that's in the trial where a modification of the established surgical intervention is to be tested. For example, if there is any established surgical intervention, if any kind of modification is done, that's also considered as a research. So any modification of the established surgical intervention to be tested is also considered as research, and the protocol must specify the need for this modification, and the expected conventional method is compared to that, and the established surgical intervention is compared with the modified surgical intervention, and the risk and benefit assessment is evaluated. And next they have mentioned that in any trial where an entirely new surgical intervention is being tested, if any entirely new surgical intervention is being tested, the ethics committee may insist some animal data, or we can say the modeling data, which establishes the efficacy and safety of the intervention based on that's a risk and benefit assessment. They have also written that the provisions for free treatment and compensation for any study-related injury must be ensured for the trial participants. If any case of during the intervention, the participants must be paid. They must get payment and any kind of injury, if they get any kind of injury, the participants must be provided provisions of free treatment and compensation. And ethics committee must determine the compensation amount after the investigation has described the relatedness. And they have also discussed about the sham surgery. That's sham surgery is like kind of a dummy surgery, like a patient a patient is given anesthesia, and there is like cutting, okay, anesthesia. uh, if you here I will write here the sham surgery. Sham surgery is a kind of a dummy surgery. Sham surgery in this, the patient is getting anesthesia. The patient is getting anesthesia, and there is also interventions like cutting, and then okay, suturing. All these things are done, but without doing any kind of manipulation, surgical manipulation. So then this sham surgery, and so they are comparing like placebo and drug is comparing here, sham surgery and a real surgery, real surgical intervention. In this, the patient is getting anesthesia, all the requirements of a surgical intervention, and there is also manipulation done. And but in sham surgery, the patient is like the patient seems to be getting surgery, but it's no manipulation is done here. So they have also written about the sham surgery. So if any, due to inherent ethical issues, sham surgery should not be included in the design of the clinical trial. So the ICMR is telling that sham surgery should be not included in the design of the clinical trial, except in cases where there is a strong scientific reason. Under such circumstances, certain conditions must be followed. The conditions, they have written that there has to be a clear description for the justification to include a sham surgery group in the protocol, which must be assessed by the ethical committee. There should be no serious harm caused by the sham surgery. The participant must get access to appropriate relevant intervention at the end of the trial. Okay. Next, they have discussed about the after sham surgery, after surgical intervention. Next, they have discussed about the community trial, that's public health interventions. So in this, they are stating that community trials or studies involving whole communities and are conducted to evaluate preventive strategies like mass drug administration trials, fortification of food studies. Such studies typically involve the whole community. The study unit could be a group, area, institute, village, block, district, and etc. And the whole population is expected to participate in the study. In such studies, different communities are randomized and allocated to different arms. Then you have suggested to read section 8 for the further details. So next topic, that is the community trials. They are discussing about the public health interventions. So they are telling the community trial, these are the studies like which are involving the whole communities and are conducted to evaluate preventive strategies like mass drug administration, fortification of food, and such studies typically involve the whole community. And the study unit, it can be either group, area, institute, village, block, district, and the whole population is expected to participate in the studies. Then next, they have discussed about the clinical trials of intervention in HIV or AIDS. So clinical trial in HIV positive patients could be for the evaluation of new drugs, vaccines, or other preventive measures and diagnostic tests. Apart from general ethical principles that apply to all clinical trials, some special issues needed to be addressed when the clinical trial are planned in patients with HIV or AIDS. Social stigma, cultural embedded myths. That is, social stigma, culturally embedded myths about HIV, marginalization, lack of legal status or criminalization of some communities that are suspected to HIV, or the disparity in the standard of care in different parts of the world are examples of such special issues. So what they are actually trying to highlight is that, for example, if clinical trial are carried out in HIV patients or AIDS patients, is carried out, why because of for the evaluation of any kind of new drugs or vaccines or preventive measures or any diagnostic tests. So there should be some specific and other general ethical principles should be applied during carrying out a clinical trial in such kind of populations. So they have written that the ethical considerations here is that social stigma, culturally embedded myths, marginalizations, lack of legal status, or criminalization of some communities that are susceptible to HIV or other disparate instead of care in different parts of the world or example of such spatial issues. Global study in HIV and AIDS in specific communities should receive approval from the relevant national authority. So they have discussed the specific ethical guidelines that the global studies in the HIV or AIDS in specific communities should receive approval from the relevant national authority. They should get approval from the relevant national authority and other relevant authorities such as HMSC where applicable, in addition to approval from ethics committee. So when testing for HIV is done, consent and pre-test and post-test counseling should be done as per National AIDS Control Organization guideline. So during testing HIV, first we need to take consent, and pre and post counseling should be done as per National AIDS Control Organization guidelines. The next issues that may arise because of discordant couple should be addressed before initiating any study in people living with HIV. So issues that arise because of discordant couples should be addressed before initiating any study in people living with HIV or AIDS. Okay. Then, as HIV is sexually transmitted disease and is potentially life-threatening, the right to life of a sexual partner must be respected. The right to life of the sexual partner must be respected over the right to privacy of the HIV positive individual. Here, that's it states that if any couple, if any one of them is that's HIV positive, then other partner should be informed, because the other partner should have the right, he or she is having the right to life. So here information should be given. Here privacy is information is respected over the right of privacy. That is, the right to life of the sexual partner must be respected over the right to privacy of the HIV positive individual. The next phase 1 studies are permissible in patients with HIV/AIDS if the drug under study cannot be tested in healthy participants due to expected toxicity. So in case of we know phase 1 studies, they are usually done in healthy patients, healthy volunteer individuals, but it is permissible to carry out in HIV or AIDS patients. Phase 1 is permissible to carry out in HIV patients if the expected toxicity of the drug is marginally high. A combined phase 1 and phase 2 or phase 2 study can be conducted in this population when other therapeutic options have been exhausted. So combined phase 1 phase 2 can be conducted in this population. When a trial with preventive HIV vaccine is conducted, it can result in positive serology. So when a trial with a preventive HIV vaccine is conducted, it can result in positive serology. This does not indicate HIV infection but can create problems for travel and employment. Under such circumstances, the project investigators should issue a certificate stating that a person in question was a participant in a vaccine trial and provide clarification on the result. For example, in case of when the clinical trial is conducted for HIV vaccine. So, for example, in case of any if the participant is non-infected, it's not infected by HIV, but to participate in the vaccine trial, then for that patient the serology can be positive for HIV. Serological tests can be positive for HIV. Then the investigator should provide a certificate to these kinds of participants that they have taken part in the trial and they are not HIV positive, like that. Okay. Research that involves sexual minorities or IV drug. Research that involves sexual minorities or IV drug users should have community engagement. So community leaders throughout the life of the project until completion and dissemination of the results. The ethics committee may also consider co-opting a member from this community if relevant for initial and continuing review of proposal. When where possible, for example, if the drug is found useful, standard of care. If the drug is found useful, standard of care is not available, or regulatory permissions are in place, the ethics committee should ensure post-trial access of the IP for the participants. For HIV positive person, any research may be misconducted. For HIV positive person, any research may be misconstructed as research on anti-HIV treatment and make them willing to participate. Therefore, the full implication in simple terms should be explained to HIV positive participants about any other research being done on them, such as research on hepatitis B. So, like you are stating, next they have done. They are discussing about the clinical trials on traditional system of medicine. So, traditional system of medicine are known for their long history, of safe and effective use. Validation of safety and efficacy using scientific and evidence-based methodology is needed for the purpose of universal accepting, gaining confidence of practitioners and satisfaction of end users in the product. So, Government of India has recognized Ayurveda, Siddha, Unani, Yoga, Naturopathy and Homeopathy as traditional Indian system of medicine. In 2012, Soba Rigpa was also added to the list. Ministry of Ayush governs and regulates the system. Drugs under this system comes under Drugs and Cosmetic Act 1940 as ASU and H drugs. Drug or formulation under this system of medicine are classified into two groups, which they have mentioned in the box 10.7 for further details. So they have classified classification of drug or formulation under Ayush. That is the first one is the classical preparations or formulations. Then second one is patent or proprietary products. First one, that's classical preparation or formulation, are those that are to be clinically evaluated for the same indication for which it is being used, or as has been described in the classical authority texts. These classical drugs are manufactured and named in accordance with the formulation described in the authority texts. Then second, patent or proprietary products or formulation containing only such ingredient mentioned in the formula described in the authority books of Ayurveda, Yoga, Naturopathy, Unani, Siddha, Homeopathy, Rigpa system, as the case may be. Medicine specified in the first schedule, but differ to create a new combination or use, innovation or invention to manufacture products is different from classic medicine. However, this group does not include a medicine which is administered by parental route. So, the next is, if IPs or comparators of one or more than one traditional system of medicine are to be investigated, the investigators from the respective system should be included in the study as a co-investigators. The ethics committee must co-opt a person with relevant expertise and expert of that traditional system of medicine to review the proposal, especially the benefits and risk of the intervention, eligibility criteria, dose of intervention, outcome plan, and traditional methods of evaluation if necessary. The next, when a folklore medicine, that is entomedicine, is ready for commercialization after it has been scientifically found effective, benefit sharing should be ensured, and the legitimate rights share of the tribe or community from which the knowledge was gathered should be taken care of appropriately when applying for the IPRS and patents for the product. While conducting trial using interventions of traditional medicine, the investigator must ensure the quality of the interventional product. The next, they have discussed on trials on diagnostic agent, then radioactive materials, then investigator initiated clinical trials. Like investigator initiated clinical trials, this is academic institutions routinely carry out investigator initiated clinical trials. In such trials, the investigator has the dual responsibility of being an investigator as well as a sponsor. So, financial arrangement must be made by the institution or investigator for the conduct of the study, as well as to pay for free management of research-related injuries and compensation if applicable. Funds should be made available, or appropriate mechanism should be established. The institution must have or introduce policies that establish mechanism to ensure quality of the data generated and safety of the intervention, such as monitoring, auditism, DSM, etc. That's data safety management board, etc. When academic clinical trial planned for off-label use of a drug, when a drug that is marketed is being used for any new indication, new dose formulation route for purely academic purpose and not for commercial use, then such clinical trial designed by researcher or academician academics may not currently require regulatory approval. However, ethics committee has to approve such studies after due consideration of benefit and risk and overall ethical aspect, and licensing authority has to be informed as per Gazetted Rule 313. Date is 2016, issued by CRESCO. The trial must be registered in CTRI also, and there should be mechanism for appropriate methods for informed consent to conduct of trial and proper follow of the patient. For student conducting clinical trial as a part of their academic thesis, so for students conducting clinical trial as a part of their academic thesis, the guide and academic institute should take up the responsibilities of the sponsor. The next, they have written clinical trials on contraceptives. So in this subsection, the ICMR is writing that several methods of contraception are available, including barrier method, hormonal method, emergency contraception, intrauterine, surgical methods. Since these studies are conducted in healthy participants, all efforts to minimize risk must be in place, and the proposed benefit must justify the foreseeable risks. The following issues must be addressed while undergoing research on contraceptives, whether they be drugs, devices, or surgeries. First, all procedure of clinical trial will be applicable. So all the procedures of clinical trial will be applicable here. For a new contraceptive method, non-comparative study can be accepted. So for a new contraceptive method, non-comparative study can be accepted. However, a sufficient number of cycles should be studied to obtain the desired precision of the estimate contraceptive efficacy. The comparator should, whenever possible, be chosen from among marketed products with a similar mechanism of action and schedule of use. In women where a non-biodegradable implant has been used, so in a woman where a non-biodegradable implant has been used, a proper follow-up for removal of the implant should be done after the trial is over or the participant has withdrawn from the trial. The educational and socio-economic level of women participants may be considered to judge whether they will be able to comprehend the use and risk associated with the particular contraceptive. Participants should be clearly informed about the alternatives available for the contraception. Any pregnancy occurring during a contraceptive trial should be followed up for final outcome to mother and child. So any pregnancies occurring during a contraceptive trial should be followed up for final outcome to mother and child. Children born due to failure of contraceptives and studies should be followed up for any abnormalities if a woman does not opt for a medical termination of pregnancy. Okay, a compensation policy must be established at the beginning of the trial to provide a cover for this contingency or issues related to trial. The next they have given about pregnancy and clinical trials. So any clinical trial conducted in women of childbearing age raises ethical issues that needs to be addressed. Similarly, studies conducted in women who are pregnant needed to be evaluated with care and ethical issues addressed. When clinical trials are conducted in women of childbearing age, they must be counseled. To use effective contraceptive methods. When a clinical trial conducts in women of childbearing age, they must be counseled to use effective contraceptive methods. These must be stated in the ICD, and it should be ensured that these methods are understood and followed by the woman participant. In clinical trials that include women of reproductive age, there may be occasional inadvertent pregnancy. In such an instant, the woman should be withdrawn from the study. And so in clinical trials that include a woman of reproductive age, there may be occasional inadvertent pregnancy. In such an instant, the woman should be withdrawn from the study, and efforts should be made to collect data on the drug effects as well as outcome for both mother and fetus. The follow-up plan of pregnancy and care of fetus must be stated in the protocol and ICD. Ethics committee to review the need if during research participation the female sexual partner of a male participant gets pregnant. The protocol and ICD must state a plan to document this, and both pregnant partner and fetus must be followed out for research and reported. Pregnant women who have right to participate in clinical research relevant to their healthcare needs, such as gestation, diabetes, pregnancies. So pregnant women have the rights to participate in clinical research relevant to their healthcare needs, such as gestational diabetes, pregnancy, induced hypertension, and HIV. So risk-benefit assessment must be done at all the stages of both the mother and fetus. Research involving pregnant women and fetus must only take place when the object of the research is to obtain new knowledge directly relevant to the fetus, the pregnancy or lactation. The criteria described in box 7.8 must be fulfilled. The criteria is appropriate studies on animal and non-pregnant individuals should be should have been completed. If applicable, the risk to fetus must be the least possible risk for achieving the objective of the trial, including when the purpose of the trial is to meet the healthcare needs of the mother or the fetus, or the risk of fetus is minimal. Researchers should not participate in decision making regarding any termination of pregnancy. No procedure change will cause no procedural changes will cause great no procedure changes which will cause greater than minimal risk to the woman or fetus will be introduced into the procedure for terminating the pregnancy solely in the interest of the trial. So women should not be encouraged to discontinue nursing for the sake of women should not be encouraged